Good day viewers and welcome to my channel. Today we are going to quickly revise some questions that are likely to come out in your biology WAEC examinations practical. So it's just a preview to questions that we're expected to see when we write our biology practical exam 2026. Now these are the specimens. You have groundnut pods containing seed. You have crushed or meal roasted groundnut seeds. Beaker with 20 ml of water. Beaker with 20 ml of table salt solution. Beaker filled with sandy soil. Portion of alimentary canal of a small mammal from esophagus to end of stomach. Then you have twig or branch of a plant with mistletoe or colored picture of a parasitic plant, evaporated milk, fresh fish, dried maize grain, tomato paste, fresh tomato fruit, rotten tomato fruit, thermometer meter rule, and quadrat. So let's get right to it. Now, specimen A, groundnut pods containing seeds. Specimen A, groundnut pods. Now, this is the taxonomic classification of specimen A. Most times, WAEC questions, they focus on the kingdom, the division, and the class. So this, of course, you have Plantae, and of course, the genus and species Arachis and Hypogia. Now, what is the botanical name of specimen A? From the last slide, you saw that the generic name is Arachis, and of course, the specific name is Hypogia. Agricultural classification of A is legume, and the life cycle an annual plant. What type of fruit is A? It is a legume. It's a pod. 2 reasons for your answers. Why is it a legume? That's what we are saying here. Because it develops from a single carpel or is split along 2 sutures when ripe to release the seeds. That means it dehisces along 2 sutures to release seeds. Then, of course, the type of germination which it undergoes is hypogeal germination. Despite the fact that it's a dicot, it undergoes hypogeal germination. Name one class of food present in A, fats and oils. You can list the other ones as well. Then name the area of the seed or specimen A where food is stored, and you have the cotyledon. So list 2 uses of specimen A. It's a source of food, and of course a source of raw materials for production of things like groundnut oil, groundnut cake, etc. Then state 2 observable differences between specimen A, the pod, and the seeds inside. Of course, the pod is light brown in color. The seeds, they are pinkish in color. The pod is rough and has a fibrous texture, while the seeds inside is smooth and have a papery texture. For economic importance of specimen A, cover crops to prevent erosion, organic manure to the soil, fodder crop for farm, and of course used in crop rotation for maintaining soil fertility. Specimen B is crushed milled roasted groundnut seeds. So what questions do we have here? You will be asked to carry out a food test. And of course, notably, the first one, you crush that specimen against a filter paper and view it against the source of light. This is called the translucent test. Now, what you will observe when you finish crushing the groundnut against the filter paper is that it will be translucent, allowing a minimal amount of light to pass through. That means by translucence. And of course, we know that fat and oil is present. So apart from other tests for fats, you have the Sudan 3 test, which or osmic acid test to detect the presence of fat and oil. Then we can also, since we are making provision for copper sulfate and sodium hydroxide, you can carry out this test by adding sodium hydroxide to specimen B in a test tube and adding copper sulfate. A violet color or bluish violet color or indicates the presence of proteins. However, there is a very low chance of them bringing out this question, but anything can happen. So then you have the other one described there with Benedict solution. Then of course specimen C is beaker with 20 ml of water, specimen D beaker with 20 ml of table salt solution. So you have two specimens that we're going to talk about side by side here because of the nature of the experiment. So what are the uses of specimen C? Helps to quench thirst, aids digestion, helps to maintain homeostasis, that's the constant internal environment, serves as a lubricant in joints, regulates animal body temperature. So these are a few of the uses of water. Alright, so name the elements. Of course, it's hydrogen and oxygen, H2O. State three characteristics of specimen C. It is colorless, it is odorless, it is tasteless. Now, this is the focus. Now, take a seed from specimen A. So we are assuming that they are going to use the groundnut seed to carry out this experiment. Take a seed from specimen A, put it in specimen C, and a seed from specimen A and put in specimen D. So it means insert a groundnut seed inside the beaker of water, inside groundnut seed inside the beaker of table salt. Then record your observation. Now you notice that the piece of groundnut inside water becomes turgid because it gains more water. Water goes inside the seed, while the piece of groundnut seed in D becomes flaccid. That means it loses water because it's a salt solution. So the piece of groundnut seed becomes flaccid since it loses water to its surrounding. Now, the biological phenomenon observed here is called osmosis. Describe what happens to specimen A when immersed in specimen D. So we have to describe the result we just got from that experiment. Now, the cell will lose water by osmosis, which is plasmolysis. The salt solution has a higher solute concentration than the cell content. So water moves out through the semi-permeable membrane down the concentration gradient. The elements present in salt water, these are some of the elements. Now, the effect of concentration of the salt solution on the rate of the biological phenomenon, or on the rate of osmosis. Then, of course, these 2 points that we raised here. The higher salt concentration in D creates a steeper concentration gradient, which increases the rate of osmosis. So, rate of osmosis is increased. Then, the greater the difference in water potential between the cell and the solution, the faster osmosis occurs. State 2 functions of specimen D for nerve and muscle functions and seasoning and preservatives. So specimen E, of course, the beaker filled with sandy soil. Pour half of, now there's a very low chance of them setting this question, but we'll revise it all the same. So pour half of specimen E or a part of specimen E into specimen C water, stir the mixture for some minutes and record your observation and conclusion. Then this is the observation you will get. However, since it's 20 ml of water, this particular question may not necessarily be asked, but we learn it all the same. Bubbles of air is seen rising from water and escaping at the surface. Soil contains air. Title of experiment to show that soil contains air. Components of composition of soil: humus, air, mineral matter, water, living organism. Please feel free to pause the video at any time. Feel free to pause the video so that you can write up what you need to write. Observable characteristics of the soil. It is reddish brown in color. They are sharp to feel when rubbed between the palms. Its particles do not aggregate together even when wet. Low capillarity, large particle sizes, large pore spaces between the particles. Then another one you can do, fill 2/3 of a beaker with sand, add water until it is well above. Shake the mixture and allow it to stand on the table for 10 minutes without disturbance. Observe the content carefully, state your observations. So this is what you observe. Again, because of what we are providing, then this is a plausible question, but because of the provisions, 20 ml of water, you can't necessarily pour it and mix it with the volume of sand we are giving. However, we can't take any chances, so we have to cover all our bases. So some light materials are seen floating on the water. Water on top looks cloudy with suspension. A layer of fine particles, a layer of coarse sand follows, a layer of pebbles and gravel at the bottom. So this is just for soil texture of sand. Specimen F, portion of alimentary canal of a small mammal from esophagus to the end of the stomach. So what class of food is digested in the stomach? So we focus on the stomach. The food digested, protein digestion begins in the stomach. Two enzymes present, you have pepsin and you have renin. You have pepsin and renin. There are two enzymes produced by the gastric juice of the stomach. Now, the enzyme is specimen F, which helps in digestion of specimen H. Specimen H is milk. So, which enzyme helps in the digestion of specimen H? The enzyme there is renin. Since renin, we just said that renin acts on casein, which is milk protein. Make a label diagram 8 to cm long of F. That means from the esophagus down to the stomach. So if we label it, you see the esophagus labeled. You see the cardiac sphincter, the stomach, and the pyloric sphincter. Now you must write the title of the experiment. So diagram of specimen H, or in bracket you can write showing esophagus and stomach. Then your magnification is written at the side, ×1, ×2, ×0.5, depending on your image size and the actual size of the specimen before you. What gland produces the enzymes named? What gland produces renin and pepsin? Gastric gland. What type of acid is present in specimen F? Hydrochloric acid. Two features that adapt the enlarged part of specimen F to its function, that's the stomach. The muscular walls for churning and mixing food with gastric juice. Stomach lining secretes mucus to protect it from self-digestion by hydrochloric acid. Now, state the functions of any four of the labeled parts in that previous diagram. We're supposed to ask this immediately after the diagram. Esophagus transports food from the mouth to the stomach by peristalsis. Remember, esophagus has smooth muscles, so food moves down the esophagus by peristaltic movement. Then the cardiac sphincter now controls the entry of food from the esophagus into the stomach. The stomach kills bacteria present in food for temporary storage of food. It churns food, secretes gastric juice, which has pepsin and renin, which aid in digestion of proteins. Then your pyloric sphincter controls release of chyme. It's CHYME from the stomach into the duodenum. What's the mode of nutrition of organisms which possess this particular specimen? It's holozoic or heterotrophic mode of nutrition. Mechanism of feeding is grinding mechanism. Okay, we asked this question before, and of course renin helps in the digestion of milk. Then you have your twig or branch of a plant with mistotol. That's what is displayed there. That's your specimen G. So you have two organisms here. You have the twig, and of course you have the mistotol, and it's a parasitic relationship. It's a parasitic association. These other types of association, we know mutualism, we know commensalism, but this one is parasitic. How does it work? The mistletoe benefits by absorbing water and mineral salts from the host through ostoria, while the host is harmed, loses nutrient, and may weaken or die. So that's a specimen G. Harmful effects of mistletoe: weakens the host by withdrawing water, mineral salts, and organic nutrient, causes stunted growth and reduced yield of the host crop, and can lead to the death of the host if the infestation is severe, because it absorbs all the available nutrients to the detriment of the host. Two ways of controlling it: physical removal and use of herbicide. So you have physical control, you have chemical control. Three other parasitic plants we know: you have your doda plants, you have your striga, you have your witchweed, you have broomrape, and the other ones. So all of them, they are all parasitic plants. Specimen H, my favorite, evaporated milk. Now we carry out this test. You add iodine solution to specimen H, and of course starch is absent. You add sodium hydroxide to specimen H and add copper sulfate. You have a purple or violet coloration to show protein is present. If you rub H against a filter paper and allow it to dry, it's going to show a little very faint translucent spot to indicate that fat or lipid is present. Then if you add specimen H to Benedict solution with heat, you have a brick red precipitate to show that reducing sugar or lactose is present. Now, the second test that was carried out above is a Buret test for proteins. Other tests that can be carried out on proteins include Millon's test, xanthoproteic test, and hydrogen test to assay for the presence of protein. Food substances that we can carry out this test on: you have egg albumin, soybean, etc., etc. Now, alternatively, because we are using 20 ml of water, they can carry out an experiment on diffusion, in which you just drop like 2 spoons of milk and record the time it takes for the milk to completely disperse in water. So this was a past question some years back on a particular specimen. So the medium of spread is water, liquid, and the sense organ involved is the eyes. It's very important here because we are going to use this information to compare with another of the specimen that was given. Fish. Kingdom, phylum, Animalia kingdom, Chordata phylum, and class Pisces or Osteichis. Now, reasons for us saying that it belongs to class Pisces: presence of scales, paired fins, operculum or gill cover, etc., etc. Its habitat is aquatic habitat. Very easy to know that. External features that adapt specimen J to its environment: streamlined body, muscular tail, counter shading coloration. Then it has gill cover, it has lateral line for vibrations, it has dorsal fins, and of course caudal fin. You can see here the title of this diagram and the magnification, though I doubt you have ×1 here. It could be, can be times 0.5 depending on the size of the fish you use and the size that you specify to draw. Methods of preserving fish: refrigeration, drying, salting. Classes of food: it has proteins, it has lipids, it has vitamins, it has minerals. Economic importance: source of food, source of oil for medicinal purposes. Sorry, I'm trying to be as brief as possible. I don't like long videos. Specimen K, dry maize grain. Is the Z maize. So dry maize grain is Z maize. Describe specimen K under the given headings. Of course, an annual, it's a cereal that goes hypogeal germination, and it's a caryopsis, and food is stored in the endosperm. As opposed to groundnut, yes, food is stored in the cotyledon. In maize, food is stored in the endosperm. So we'll quickly carry out this test. Add iodine solution to the cut section of specimen K, and you see that a blue-black color is noticed. Blue-black coloration is noticed, and that's to tell you that starch is present in maize. So though some parts will not change, so in the parts that don't change color, starch is not present, but the endosperm itself, the endosperm area, will turn blue-black to indicate that starch is present. But you can write just turns blue-black, no need to put the endosperm there. It still signifies that starch is present. Of course, this was one of the past questions some years back, and of course you see the magnification times 5, which means it's far larger than the actual drawing of the specimen. This is specimen L, tomato paste in a petri dish. Describe how specimen L is produced. This is the way it's produced: washed, blanched, crushed, strained, and of course sealed in cans or jars. 2 ways of preserving this: by canning, by sterilization. Fresh tomatoes. In fact, we are working with a lot of tomatoes, both fresh, rotten, and all of them sachet. Now, this is the classification of tomatoes. Remember, it's Solanum lycopersicum. Solanum lycopersicum. Describe it's an annual. It's a vegetable crop agriculturally, and of course, it is a berry. It is a berry. What type of placentation? Axial placentation. Why? Because the ovules are attached to a central axis within an ovary formed by fused carpels. So that is why it is axial placentation. Other fruits with similar placentation: orange, garden egg, grape, lemon, etc. Rotten tomatoes. It stinks just from the screen here. Now, rotten tomatoes in a petri dish. What are the reasons it's appearing like that? Pest infestation, poor preservation of the tomato, inadequate transportation, which makes it to spoil quickly, untimely harvesting of the specimen, inadequate storage facilities. 3 ways damage could have been prevented: proper storage, proper pest control, and moderate temperature. Now, what are the differences between specimen M and N? That's the difference between the firm tomato and the rotten tomato. This one is firm and turgid. The other one is soft and breaks easily. This one is pleasant to the eyes and has a mild fruity odor. The other one has a very foul, offensive smell. So it has a smooth skin. This one, the skin is not too smooth. It has a lot of lacerations. Then the bright red. This one is dull or darkened or discolored. This one, the cell walls are intact, while you have broken cell walls. While this one is free of bacterial colony, this one shows bacterial growth. Now, cut open specimen A. This is like a follow-up to that question where we dispersed milk in water. So this time around, just slice specimen N open and perceive the odor. And of course, it takes about 1 to 5 seconds for you to perceive the odor. And what has happened is diffusion has taken place. Medium of spread is air, and of course the nose is used to perceive it. If you notice, the time for the diffusion in air is faster than that in liquid. So see, air is a faster medium of diffusion than liquid. Thermometer, that's specimen P. So state the use of specimen P to measure temperature. And we know temperature is the degree of hotness or coolness of the body. And the unit of measurement of this particular ecological factor determined is Celsius. So temperature is measured in Celsius or centigrade. The liquid element in specimen P is mercury. What are the precautions taken with specimen P, especially in studying the temperature of habitat? So at least 2 readings in a particular area are taken to ensure accurate result. The type of thermometer that is used to record highest and lowest temperature of the day, maximum and minimum thermometer. This is specimen Q, meter rule, or what we know as ruler. State the use of specimen Q. Specimen Q is used to measure the depth of water in ecology and to measure the length of organisms or habitat. How is it used? Now, a weighted line is attached to the end of the meter rule to ensure that the rule is vertical. It is inserted into the body of water. List other instruments for measuring ecological factors and what they are used for. Barometer for air pressure, rain gauge, rainfall, photometer for light intensity, wind vane for direction of wind, anemometer for velocity of wind, circuit dish turbidity, hygrometer relative humidity. Specimen R is a quadrat. Specimen R is a quadrat. What's the function of specimen R? It is used for population studies, determining the population of small organisms and plants. And of course, describe it. It's a square or rectangular frame made from thick wire. Another method of collecting sparse specimen, in addition to the quadrat method, is the transect method. Describe how specimen R is used. It is thrown at random several times, and on each landing, the area covered by it is observed. The type of species and their number within the quadrat are recorded. The average number of times each species appears is calculated, and the most frequent or dominant species is determined. Name some organisms associated with the use of R. You have Tradux, Botalinum, you have speargrass, you have beetle, all these smaller animals that you can throw and they can still possibly remain within the quadrat. But majorly plant species, since they don't move, so it is very easy to check them. Now, name the area of biology where specimen P, Q, and R are used. Yes, the thermometer, meter rule, all of these other instruments we named, and the quadrats are used. So they are used in ecology. Population studies in ecology uses all of these instruments that were named. So guys, thanks for watching. I wish you good luck in your examinations. Don't forget to subscribe. We're going to have intense discussions on different biology questions and their possible answers. Thank you.